Sure. Okay, so I'm going to talk about GWAS as a window to genetic architecture in a couple of different ways, and I'd just like to say thank you for the invitation to present from the organizing committee and the chairs. And I'll talk a little bit about polygenic prediction and heritability and some of the emerging analyses that are coming from the work as part of the Psychiatric Genomics Consortium. And then I'll describe Mendelian randomization. And I think MR is an excellent example of something that you can do with just the summary results from the kind of GWAS catalog in some of the secondary traits. And we'll talk through some examples of that more precisely. So to kind of get things, sorry about that. To get things started, a few years back, I guess it's probably almost 4 years now, the International Schizophrenia Consortium, led by, kind of Pamela Sklar and efforts from Sean Purcell and Peter Vischer, used polygenic prediction, this idea of taking lots of individual SNP associations and summing them up to predict risk to disease in a different population or a different sample. And these series of papers kind of initiated a new round of activities in GWAS more particularly of trying to understand, whether there is kind of a genome-wide contribution to complex traits from common variants more generally. And you can read about that in the kind of various papers presented on the slide, and also the more messagey paper from Naomi Ray and Mike Goddard and Peter Vischer, in terms of kind of some of the conceptual underpinnings of this. But at its very core, this idea that we have multiple small genetic risk factors that are being captured by genome-wide association data, and that those sum up together to create this kind of underlying liability is a nice kind of reconciliation with the kind of biomedical model and old quantitative genetic theory from Fisher and Sewell-Wright and others going back almost, I think, over a century now. And these kinds of methods and methods in this vein, so on the next slide, we have this Bayesian inference analysis of the polygenic architecture of rheumatoid arthritis that Eli Stahl led, where it took a similar sort of concept. So it's again taking this polygenic prediction, but kind of trying to evaluate over improving levels of prediction as you move further and further, down your list of associated variants. How much better do you do in terms of prediction, and what can you say about genetic architecture on the basis of that? And you can make some estimates at the number of kind of associated SNPs for various diseases. So Eli worked primarily on autoimmune disease, and so that's why you see rheumatoid arthritis and celiac disease as the kind of two primary considerations. But this was also applied to myocardial infarction and coronary artery disease, as well as type 2 diabetes, showing that there exists, and I think this is consistent with all of the data that we've generated thus far, somewhere on the order of thousands, maybe even tens of thousands of variants that are associated to these complex traits in the population more generally. So that kind of long tail concept is very much a reality when we think about the genetic architecture of complex traits. You can go the other direction and just take the very top end of associations. So this is work that Chris Katsakis led with Mark Daly and others in the autoimmune disease community. And this paper in PLOS Genetics took the attitude of the top loci emerging from the genome-wide association results of various individual autoimmune disorders, including things like rheumatoid arthritis or MS or lupus and so on and so forth, that there were patterns of association that were emerging across those diseases, that you saw the same variants being associated to multiple different diseases, or the same regions of the genome being associated to multiple different autoimmune diseases. And that actually not only suggested some biological overlap between these phenotypes, but also could be used to help parse out the underlying biological mechanisms. So you can contrast those sets of results that group together genetically with those sets of results that group together from protein-protein interaction data. And you obtain this, kind of further insight into the kind of structure of the underlying biology across these different diseases. And any kind of overall discussion of genetic architecture would be remiss without talking about GCTA. So this is a package that Zhang Yang and Hong Li have been working on with Naomi Ray and Peter Fisher and Mike Goddard in Australia. And there are a series of papers in Nature Genetics and I think in Nature as well, describing the overall concepts here. But basically, the general principle from a GWAS perspective is that if we have a trait that is heritable, the amount of genetic similarity that you observe in the population should correlate with the amount of phenotypic similarity that you have. That's a very simple and attractive idea, but it is worth mentioning that any artifact that you have in these circumstances will yield a bias in your estimate of heritability more generally. But they've extended this work more recently, so you can see the paper published in Bioinformatics, talking about this conceptually, and I'm going to describe a little bit of the work that's being done as part of the PGC, the psychiatric GWAS meta-analysis effort, to try and take this into a bivariate direction. So the idea here is that we now have the opportunity to be able to both estimate the heritability within phenotype, but also estimate the overall genetic correlation between a pair of phenotypes. And that's very important in a number of different domains. In particular, in scenarios where you have rare phenotypes, or in scenarios where you have a large, ascertained population of cases that it's difficult to get epidemiological surveys of these things more appropriately, or even to do the kind of bivariate or multivariate heritability analyses in families from genetic epidemiology because of the frequency of the disease and the challenges associated with collecting family members for said disease. So if it's a late onset disease, for example, then you are going to have a hard time to be able to really pick off, parents and grandparents and so on and so forth for that given phenotype of interest. But again, the same caveats apply in terms of correlated artifacts being problematic. Nevertheless, this is work that we've put together in the context of the PGC and has just been accepted at Nature Genetics. And again, this has been led by Naomi Ray and Hong Li and Ken Kendler. And what you can see is that the kind of on the left here, these are the univariate estimates of SNP heritability. And what you see is you see strong heritability estimates, kind of 20 to 25% of the variance for each of the primary 5 diseases of ADHD, bipolar, schizophrenia, major depression, and autism spectrum disorders. So again, this gives us the sense that there are genome-wide significant variants to be found if they perhaps haven't been found yet. And then second to that, you see strong overlap, in particular between schizophrenia and bipolar and major depression, and schizophrenia and major depression and ADHD. This is the kind of significant line on up in terms of those estimates of not only SNP heritabilities, but bivariate correlations. And then you can look at the kind of individual level SNP correlations that are standardized against the total heritability. And what is revealed is that the SNP correlation between, say, bipolar and schizophrenia is approximately 70%, which means our estimate at this point is half of the genetic variance in the population attributable to schizophrenia is shared with bipolar disorder. And that's a really important insight emerging, not only in terms of the nature of the risk for these diseases, that it's going to be, there are common variants to be found, and that these GWAS activities will yield significant findings, but also that our phenotypic characterization as it stands now does not actually accurately reflect the etiological basis of these diseases. Now, there are specific effects as well, so that, this is kind of an active area of research and things that we're interested in particular for things like psychiatry, where our ability to kind of access biomarkers and tissues and so on and so forth is quite poor by comparison. So these, results are, some of them are publicly available, so I've kind of highlighted other places where the genome-wide association results, rather than just significant loci, have been identified as being put online, but also the full results, because I think there is value, in a sense, in both activities. If you're trying to filter through a whole bunch of different phenotypes and look at, kind of specific individual locus sharing, going and downloading each of those individual data sets is going to be difficult, and the kind of GWAS catalog can certainly facilitate that. But if you want to take a deeper dive view of a specific phenotype, then having the full results available as well, I think, is a really important contribution, and that's what Ricopoli does for some of the results files that we generate at the Broad as part of the PGC and other collaborative endeavors. Also Enigma, which is the kind of structural MRI Genome-wide association kind of meta-analytic consortium. They, publicly serve up their results as well. Similarly, the educational attainment GWAS. I think Nancy's comments about GTEx are also similarly the case. So there's this general move, I think, to try and put all of these results online because they really are valuable and they can help you interpret a specific phenotype. But in terms of aggregating across the entire landscape of phenotypes, I think there's certainly a value in the context of the catalog. So we're learning things about heritability, a little bit about biology, something about phenotypic structure and those sorts of things. And I think the kind of implications of the work thus far is that as we continue these GWAS activities and we continue to improve the sample sizes and increase the quality of the genetic variants that we're analyzing, that more genome-wide significant results will emerge. I think that's a pretty safe bet. About as safe as the link between, say, smoking and lung cancer. And not only does our ability to identify these variants improve, but also so too will our predictive ability. So there will be improved kind of genome-wide prediction methods and applications to a bunch of different outcome metrics. I think some of the challenges in terms of managing this is how the GWAS catalog is going to manage multiple iterations of meta-analysis, because this is kind of more emerging trend more generally in the community of what is the strategy going to be for, PGC2, for example, will take everything that's in PGC1 and then bolster it. And so how you reconcile that and update that, I think, is something of an important question. And also, the kind of, whether it wants to become a repository for these full results as well, or at least link out to them, I think is another thing that should be in the framework of discussion. But what we can also do with these results in kind of downstream analyses, somewhat similar to Chris Katops's paper, is try and understand the nature of the relationship between some of the traits that we're measuring and additional phenotypes of interest. So Rondo, Mark Daly, and say Catherine and the Global Lipids Genomics Consortium have been, we've been working together to try and understand the nature of the relationship between plasma lipid levels and myocardial infarction. So, generally at a kind of conceptual level for this particular kind of cherry, as it were, we see an increase in LDL cholesterol being associated with increase in risk of CAD. This is just epidemiologically, it has nothing to do with the genetics at this point. Decreasing HDL is associated with decreased risk to CAD, and increasing triglycerides is associated with increased risk to coronary artery disease. And there's a paper in JAMA that kind of takes a look at each of these different lipid fractions and says, we see these correlations very clearly, and there's the triglyceride observation of epidemiologically we see this increase in triglycerides, we see an increased risk in heart attack. So if we control for additional risk factors, and those additional risk factors include the other lipid fractions as well as things like smoking and BMI and so on and so forth, that really kind of flatlines the signal with respect to triglycerides in terms of its predictive ability. So most of its predictive ability from an epidemiological point of view is captured by modeling all of the other risk factors that we know. Now, that doesn't necessarily say anything about the specific mechanistic action. It just means that the phenotypic correlation is sufficient to explain most of the increase in risk that we observe over time. And we have a harder time with that for HDL and for non-HDL cholesterol more generally for what it's worth. So the big limitation here is that it does not actually imply causality or even the lack of causality. Just because I can control for the increase in triglycerides and flatline it with respect to the increase in risk to CAD does not mean that I have the ability to actually kind of state unequivocally that triglycerides are not causally related to coronary artery disease. And this is further compounded by the fact that there's a pretty nasty phenotypic correlation between triglycerides, LDL cholesterol, and HDL cholesterol, where HDL and LDL don't show much in the way of correlation at all, but you see somewhere on the order of 20 to 30% correlational structure between triglycerides and the other two lipid measures. And so that makes for some difficult interpretation. So there's this concept of Mendelian randomization, and what Mendelian randomization is trying to do is, in the same way that we have a randomized kind of clinical controlled trial where we, assign out, treatments to different groups and then evaluate what the outcomes are, the concept of Mendelian randomization is that genotypes have been assigned at random in the population, and we can similarly evaluate what the outcome is for being exposed to one allele versus another allele. Now, it is essential that you balance confounders between groups. That's true for clinical trials. It's also true for Mendelian randomization, so things like principal components and so on and so forth are really important to think about in these sorts of analyses. But then you can compare these outcomes, and that gives you what the MR people call a instrument to kind of test the validity of these sorts of things. So, one very clear example is variation in the LDL-R genes. This is the LDL receptor that has an impact on LDL cholesterol. There's a downstream impact of LDL cholesterol on coronary artery disease, and you see clear association between the LDL-R variation in the LDL-R gene and coronary artery disease more generally. It doesn't mean that the other things like smoking and nutritional status and all that kind of stuff isn't having an impact, but we see this association both to the intermediate phenotype and the downstream phenotype very, very clearly. So Ben Voigt and Sake and Gina Peloso and others wrote a paper that's in the Lancet that really describes the nature of the relationship with HDL cholesterol, because this has, again, been epidemiologically demonstrated to be associated to risk to coronary artery disease. But the question was still whether or not there was a mechanistic relationship. So that LDL-R example gives us confidence that LDL cholesterol is mechanistically related to the generation of coronary artery disease. But it was unclear for HDL. We have this epidemiological correlation, and here you can take individual associations that have very, very genome-wide significant results on plasma HDL levels and ask the question about what is the risk of that SNP to MI more generally in the population. And as you can see, the meta-analysis lines up with an odds ratio of 0.99, pretty miserable p-value in the scheme of things from, the kind of, whole bunch of different studies collapsed together. So this gives us some confidence that the mechanistic link doesn't actually exist between HDL and heart attack, but that it's actually perhaps indexing something else, that it's correlated with something else. And we can do similar things in terms of looking more specifically at the odds ratios for the increase in plasma lipid level for LDL cholesterol and what kind of p-value you get when you do a genome-wide association for that same SNP or a set of SNPs, a kind of composite polygenic score on myocardial infarction in the context of LDL. Building a polygenic score gives you a p-value of 2 × 10⁻⁻⁻. For HDL cholesterol, a p-value of 0.63. So really no kind of composite evidence that those genetic influences on HDL cholesterol are downstream associated with myocardial infarction. And so this really argues that HDL isn't actually mechanistically related to what's going on in CAD or MI. So here we have the kind of observation that LDL is related. Yes, we're confident in that. We've completed the loop in a sense. But there's this question of triglycerides, and as I said earlier, the relationship between triglycerides is very complicated by the fact that we have these correlations between the different lipid level fractions in the population more generally. So this problem is compounded not only by what is happening epidemiologically, but that phenotypic correlation that we observe is in part influenced by genetic overlap. So these are the number of SNPs that are associated with, so this is any genome-wide association SNP for each of the 3 phenotypes, and then thresholding at a p-value of 0.001 for the overlap. And as you can see, there's pretty robust overlap between both LDL, HDL, and triglycerides in terms of SNPs that are associated to one of them are also associated to others of these fractions. And that really kind of muddies the water in terms of our ability to do this kind of clean Mendelian randomization kind of activity more generally. So what we did was took the 185 top SNPs from 157 loci that have been associated as part of this GLGC effort. We've taken GWAS, this GWAS for lipids involves 180,000 individuals, and then we've taken a meta-analysis from myocardial infarction that has over 95,000 individuals. And what we're going to look at here is the effect size of each individual SNP on triglycerides, LDL cholesterol, HDL cholesterol, and coronary artery disease. So we're going to try and predict what that risk for that SNP is on coronary artery disease as a function of the effect sizes for each of the different lipid fractions. That's the general model conceptually. So we can take a couple of toy examples to see what things look like. Here are two variants that are associated to both LDL cholesterol and triglycerides, and you can see the p-values for coronary artery disease are not genome-wide significant, but they're pretty reasonably significant. And I'll bet pretty aggressively that in the fullness of time, both of these variants will be associated at a genome-wide level with respect to the coronary artery disease, in part because of the influence on LDL cholesterol more generally. So here's the two that are both genome-wide significant for LDL and triglycerides. Strong enrichment of association. If I'm going to pick two SNPs at random, I'm never going to see those kinds of p-values in the scheme of things. And then if I take the set of SNPs that are moving in an opposite direction, okay, and remember, if we do this just in LDL cholesterol, we get a pretty striking pattern. That polygenic risk score for LDL variants was pretty predictive at a kind of 2 × 10⁻⁻⁻ for coronary artery disease in a genome-wide sort of fashion. We're not really seeing any association to myocardial or to coronary artery disease as a function when the SNPs for triglycerides and for LDL cholesterol are moving in opposite directions. So we have a 10⁻⁻43rd on LDL cholesterol, that SYT7 gene variant, and 10⁻⁴ for triglycerides, and we see really no association with coronary artery disease whatsoever. So that takes us to kind of maybe a graphical representation of these things. And what you have on the x-axis here is the low-density lipoprotein, the LDL cholesterol data, and then on the y-axis here, the triglycerides data. And the dots are color-coded by the p-value for CAD. So the genome-wide, for the things that are 10⁻³ or more significant are black dots, and they tend to be clustered up in this quadrant and down over here, and are less prominent in this sort of territory overall. The things that are moving in opposite directions have more muted significance more generally. And I understand that this plot is a bit of a kind of squint and see what you like sort of thing, so maybe we can formalize it a little bit more. And the way that we formalize it a little bit more is to take the individual SNPs. We then have betas for those SNPs on each of the individual lipid fractions. So, for genome-wide significant things, they should have an alternative. Some will be distributed like they're null, and some will be part of the alternative. For the genome-wide significant loci, we have those significant impacts on LDL, HDL, or triglycerides. But we get betas for each of those different lipid fractions accordingly. And then we have this beta for CAD, for that same SNP. So we're using the same betas. And what we're trying to do is predict the beta for CAD, that's our Y in this first regression, and we're going to use the beta for LDL cholesterol to predict that, because we already saw that the polygenic score predicted well, so this is a good way of quantifying the amount of risk that you're explaining in terms of coronary artery disease. And then as a second step, we can take the residuals of this first regression and try and predict whether there is any additional information contributed by the beta from triglycerides. And this way we can be absolutely confident that there are additional effects implied from triglycerides rather than simply those effects that are being correlated and picked up by LDL cholesterol more generally. So here we have the different models, and we can do this in all iterations, and we have the various outcomes. The outcome is static. That's our effect estimate for that SNP on myocardial infarction. And then we have our predictors. So that's the various betas that we'll use, LDL, HDL, and triglycerides. And then we can include covariates in these analyses. So we can regress out the impact of some of the individual variants' impact on those other lipid fractions in an attempt to control for that potential confounding. And so when we run through this, what we see is that at the model 1, the LDL cholesterol is very significant, p-value is 10 to the minus 20. With respect to the coronary artery disease beta, so we're predicting the overall beta of coronary artery disease for this set of SNPs with a p-value of 10 to the minus 20. That's pretty good for 185 SNPs. And then if we add in HDL cholesterol, we don't see much in the way of change. And then we get this slightly curious thing of an improved p-value when we add in the data for triglycerides. And I'll come back to why I think that's the case in a moment. We look at HDL. We model HDL on its own. We don't see much in the way of we see a bit of association. We see this kind of 6 times 10 to the minus 4 sort of thing. But when we include LDL and triglycerides as covariates, we're left with this p-value of 0.35. Now, similarly, we can do triglycerides, and when we model triglycerides and we do LDL, we get this, 10, or just triglycerides for CAD, we get a 10 to the minus 8. We get 5 times 10 to the minus 12 for including LDL cholesterol. So again, that same pattern between those two phenotypes. And then the p-value down here, when we include all terms in the model, is 10 to the minus 9. So really, this is giving us a clear indication that for this set of markers, the inclusion of the effect size on triglycerides is significantly improving our ability to estimate the effect size on coronary artery disease. The reason that the p-value improves when we include triglycerides in the model and are estimating the influence of LDL is because we've shrunk the variance of the beta CAD by taking out true effects. So there are those true effects that are unrelated to the LDL cholesterol influence are counted as error in the regression without including the covariate, and then they get diminished when we do include the covariate of the triglycerides. And that's why we're seeing that improvement in the level of association between these individual betas. We can take a look at the SNPs with opposite effects on LDL and triglycerides on coronary artery disease. So here we restrict the size, the magnitude of the beta, and we really don't see any relationship between the LDL cholesterol betas and the coronary artery disease. So this is not significant when we restrict to those things. When we take things that have a moderate effect or large effect on triglycerides, but minimal effect on LDL cholesterol, so because the overlap between these loci is so great in terms of the influence, there's a lot of pleiotropy here, we have to take the tack of reducing the amount of impact, make the beta small on LDL cholesterol. And we get 44 SNPs, and there we see this, 3 times 10 to the minus 5 sort of association between that composite beta triglycerides and the downstream impact on coronary artery disease. So the kind of primary top-line conclusions here are that, GWAS is rapidly improving our understanding of the genetic architecture of complex traits. We're gaining insight into the nature of the structure of phenotypic overlap across diseases in the same or even different disease domains. We're understanding what we might be able to capture by engaging in genome-wide association, that these efforts should continue and will continue to yield further loci that will improve not only our ability to predict who will get sick, but also predict or what causes variation in the population, but also understand the nature of the biology underpinning those things. But we can also leverage the genome-wide association toolkit to really improve our understanding about specific causal mechanism. And that's one of the kind of true values of the GWAS approach, that we can start to gain insight into these mechanistic things. And so as the sample sizes continue to increase, we'll increase our the amount of genome-wide significant loci, our predictive ability, and our understanding about the nature of the overlap between these various diseases and what biological pathways may well be involved. And with that, I will close and say thank you.